Hello everyone! For today's catch-up, let's deepen our understanding of entomology. But first, what is entomology? So, to give you an initial background, it is a branch of biology that focuses on the study of insects. Knowing that insects are one of the most important factors in the transmission of most diseases, the course in medical entomology is responsible for detecting the role of insects in the spread of disease, in discovering ways of protecting food, fiber crops, and livestock from being damaged.
The most successful group of organisms on Earth is without a doubt the arthropoda, which includes insects, spiders, mites, and their relatives. Nearly 55% of all species known to science are insects alone. In our first laboratory exercise, we focus on studying and familiarizing ourselves with the characteristics that are important in the proper identification of arthropods. By watching the provided reference and drawing what we've learned, we understood that external morphology or characteristics of the insects are important in the proper identification, and things like sizes, shapes, and colors are not important in identifying them. Insects have the most developed tagmosis, meaning their body is arranged into 3 well-defined body parts known as stigmata. Their head contains the sensory organs that perceive and process external information. The thorax bears the locomotory organs, and finally the abdomen, where the organ system is located. We locate these parts by viewing them in different anatomical positions such as the lateral, the dorsal, anterior, ventral, and the posterior views.
Collecting methods may be divided into 2 broad categories such as the active and passive methods. The active method involves the use of nets, aspirators, and baiting sheets, whereas in the second method, the collector participates passively and permits pitfall dish traps and baiting to do the work. Both collection methods were done within the FEU campus. In active collection, sweeping net made from badminton racket with attached nylon net was used to capture flying insects and placed in a killing jar charged with chloroform. Killing jar is the most used method for killing collected specimens, wherein insects can be properly mounted and studied. For general hand collection, a test tube with cap containing alcohol was utilized as vial, in which minute specimens collected from hand picking will fall easily when shaken. A small fine brush with its tip moistened allows the tiny specimens to adhere to it and push them towards the collector. Baiting is a technique done for passive method. The collecting tools employed were 2 kinds of baits to attract foraging ants, including tuna bait as a protein-rich food substance and peanut butter bait as a carbohydrate-rich food substance. 20 pieces of 1/4 bond paper were used to hold the baiting food substances to attract the specimens. The baited insects were sprayed with alcohol using an alcohol sprayer in order to stop them from moving quickly because they are very agile. And 20 pieces of Ziploc served as a temporary storage for the collected specimens from the baited papers.
The third laboratory exercise revolved around basic collection of arthropods. We did sweep netting and general hand collection as active collection methods and baiting as the passive collection method. For the sweep netting, our group went to 3 outdoor sampling points around the campus, and we were able to collect a mosquito, a fly, and a moth, which were then placed in a killing jar charged with chloroform. For the general hand collection, there are 2 outdoor sampling points where we were able to collect ants and a mosquito. The ants were placed in a tube filled with ethanol, while the mosquito is placed in the killing jar. For the baiting, our group prepared a total of 10 baits, wherein 5 were distributed inside the arts building and the 5 were placed around the outside of the arts building. The baits were left for 30 minutes, and after the time elapsed, the baits were collected and placed inside a Ziploc bag. After the collection, we recorded the number of specimens collected in each sampling point. Then, in the processing of the collected arthropod specimens, forceps are used to transfer the specimen from the Ziploc bags to a petri dish with 95% ethanol and then transferred to a new petri dish with tap water, before placing them inside the test tube filled with ethanol. They were later placed in an empty slide and observed in a dissecting microscope for morphospecies identification. Then, we selected a representative for each morphospecies. In total, we collected 4 ant species, 2 fly species, a moth, and a mosquito. These specimens were mounted and we examined them under the dissecting microscope, and we captured the anterior, dorsal, and lateral view of each specimen.
The arthropod morphological characters are used for a system of classification that enables us to impart order to a complex environment. There are more than 100 morphological terms needed to be familiarized with to classify the subfamily and genus as well as the species under the Formicidae family. The collected ant specimen was identified and classified according to the dichotomous key of General and Alpert 2012 based on its distinguishing characters. First is the postpetiole, which is convenience term for what is morphologically the third abdominal segment when it is reduced in size and markedly separated from the petiole. This character is absent in our specimen. Second is the sting. It is internal in worker and queen from ant subfamilies, since it is concealed within the apical visible segments of the abdomen. This character is also absent in our specimen. Third is the acidopore. It is the orifice of the formic acid protecting system and a diagnostic tool for subfamily Formicinae. Fourth is the mandibles, an appendages with which ants manipulate their environment. And fifth is the eyes that helps ants to see light and shadows. These 5 morphological characters tells us that this ant species is from the subfamily Formicinae and its genus is Ecopila. Lastly, the Lab 4 is also composed of terminologies regarding insect viewing such as the dorsal view, ventral view, full face view, and profile view, or also called lateral view, that we use to properly view and examine the insect's morphology.
To study the morphology and the classification of the different species within the class Insecta requires great patience, understanding, techniques, keen observation, and critical thinking. And we, as the medical biology student who strives to learn about everything that lives around us for the purpose of discovery, identification, relationship roles, and health benefits, firmly believe that studying to understand is not with utmost simplicity but with the masteral of complexity.